Hello, hi, Assalamualaikum dan selamat sejahtera to all my lovely girls. Okay, today we are going to continue our discussion on cuti belajar handout. Okay, so please open your handout and get ready. Yeah, so please check your answer with me. Okay, for today we are going to discuss chapter 5. Yeah, the title of the chapter 5 from 4 is Metabolism and Enzyme. Okay, let's take a look on the question 1. Okay, question 1, diagram 1.1 shows a metabolic reaction that occurs in the living organism. Okay, so this is diagram 1.1. A substrate Q react with the water with the presence of the enzyme R that produce glucose plus glucose. Alright, so when the presence of the enzyme here, this is referred to the enzymatic reaction. Yeah, enzymatic reaction that involve hydrolysis. Okay, why hydrolysis? Because in the presence of water. Yeah, so the substrate Q is broken down into the product here, yeah, okay, with the presence of the water. Okay, so we go to the question A1. Name substrate Q and enzyme R. Okay, substrate Q and enzyme R. Okay, what is the key given to you here? It is referred to the product. Yeah, the products that produce here are glucose. Yeah, both of the product here are glucose. Okay, so what is the substrate Q? It is referred to the disaccharide. Yeah, disaccharide, which is maltose. Yeah, so maltose, when it is broken down, it will hydrolyze into glucose plus glucose. Okay, so what are the enzymes? Yeah, enzymes that are responsible. Yeah, responsible in the breaking down of this substrate. It is referred to the maltase. Yeah, maltase. Why maltase? Because the substrate is maltose. Okay, so the enzyme name is right after the substrate here. Okay, if the substrate is maltose, so the enzyme name is maltase. Yeah, if the substrate is a sucrose, so the enzyme name is sucrase. Okay, do you still remember? Alright, so that is the answer for the A1. Substrate Q is maltose, enzyme R is maltase. Okay, next, we go to the question A2. State the type of metabolism in the reaction. Yeah, the type of metabolism. So we already learned that there are two types of metabolism in our body. Okay, what are they? They are catabolism and anabolism. Yeah, okay, catabolism refer to the breakdown process, yeah, whereby the anabolism refer to the build-up process. Alright, so in this diagram 1.1, so this is referred to the catabolism. Okay, why catabolism? Because substrate Q are broken down into the product here, yeah, into the product. So this is referred to the catabolism. Okay, next we go to the question 1B, 1 and 2. Okay, let's go to the question first. Diagram 1.2 shows the stages in the enzyme R reaction based on the log and key hypothesis. Yeah, log and key hypothesis. So this is referred to the diagram 1.2. Yeah, all right. So you have to observe, yeah, the diagram 1.2 carefully. Yeah, okay. You are given here two type of substrate Q and substrate S, and there is one enzyme which is referred to the enzyme R, and this is referred to the enzymatic reaction that occur here. Yeah, and then will produce the product here. So this is referred to the enzyme reaction based on the log and key hypothesis. Okay, let's go to the question 1. Explain why the enzyme R only acts on substrate Q and not on substrate S at stage 2. Alright, so this is referred to the stage 2 as we can see from the diagram. So the substrate Q, the substrate Q acts, or bind with the enzyme R. Okay, so the substrate Q bind with the enzyme R here. Right, so the key word here, you have to look at the key or the clue given here. Okay, the enzyme R reaction based on the log and key hypothesis. Okay, log and key hypothesis. So why the substrate Q, or just the substrate Q only bind with the enzyme R? Okay, so what is the reason? It is referred to the it is explained by the log and key hypothesis. Okay, let's go to the suggested answer here. Okay, point 1, enzyme have specific active sites. Okay, 1 mark here. Enzyme that is referred to the enzyme R here. Enzyme R here have a specific active site. So this is referred to the active site of the enzyme. Okay, every enzyme have its own specific active site. Alright, so the active site of the enzyme only fit with a certain substrate. Yeah, only fit with a certain substrate. Alright. Okay, next we go to the point 2. Substrate Q has a shape that complement or fit to the active site of enzyme R. Yeah, substrate Q have a shape that complement, to the active site of enzyme R. Okay, second mark. Then point number 3, the enzyme can bind with substrate Q. Okay, the enzyme R here can bind with the substrate Q. And point number 4, substrate S does not complement to the active site of enzyme R. Okay, substrate S here is not complement. Okay, why not complement to the active site of the enzyme R? Because it has different shape. Yeah, different shape that not complement with the active site of enzyme R. Alright, and then point number 5, enzyme cannot bind to the substrate S. Okay, so this is referred to the suggested answer. So the full mark here is 2 mark. Right. Okay, next we go to the question 2. And B2, enzyme R is only required a small quantities in the reaction. Okay, so explain why. So required in small quantities is the characteristics of the enzyme. Yeah, so the enzyme is only required in small quantities, in interaction. So you have to explain why. Yeah, why we just only need a small quantities of the enzyme in enzymatic reaction. Okay, so what is the reason? Okay, we go to the suggested answer here. The structure of enzyme remains unchanged, right? Yeah, the structure of enzyme remains unchanged after reaction. Okay, then the enzymes are not destroyed at the end of the reaction. So the enzymes are reusable. The enzymes are reusable. Okay, so why the enzymes are reusable? Because the structure of enzymes or the active site of the enzyme remain unchanged after the reaction. And the enzyme also are not destroyed. Not destroyed at the end of the reaction. Okay, so that is the answer for question 1B1 and 2. Okay, now we go to the question 1C. Excessive intake of citrus food, citrus food will affect the action of enzyme R on its substrate. Explain why. Okay, so first of all, you have to know what is a citrus food. A citrus food refer to the sour food, makanan yang rasanya masam, Citrus refer to the food that contain high vitamin C. High vitamin C. For example, lime, orange. Alright, so it is referred to the citrus fruit. Okay, so excessive intake of citrus food will affect the action of enzyme R. Okay, so how does the citrus food affect the reaction of the enzyme are. Okay, so explain why. Now we go to the point 1. The pH of the medium becomes low or acidic. Okay, so we have to know that. Okay, the citrus fruit, citrus fruit has a sour taste, right? So sour taste because the pH of the food is low or acidic. Okay, so it will cause the pH of the medium becomes low or acidic. And then what happened? What happened when the medium, when the enzyme become in the medium of the acidic, so it will cause the charge on the active side of the enzyme has changed. It will cause the change of the charge on the active side of the enzyme. Okay, next, point number 3. So the enzyme substrate complex cannot be formed. Enzyme substrate complex cannot be formed. And last point is enzymes are unable to react with the substrate. Okay, so this is occur when the excessive intake of the citrus food. Citrus food. So this is referred to the how the citrus food affect the action of enzyme are on the substrate. Okay, so alright, so that is the answer for the question 1C. Okay, let's go to the question on the section B. Okay, we go to the section B, which is essay question 1A1. Okay, 1A1, diagram 1.1 shows a graph of the effect of temperature on the pepsin enzyme reaction. Okay, so please underline here, effect of temperature on the pepsin enzyme reaction. Okay, next, okay, we go to the diagram first. So this is referred to the graph. Okay, graph that show the effect of the temperature. Temperature refer to the MV, manipulated variable here. So the RV refer to the rate of the pepsin reaction. Okay. So this is referred to the point P, point Q, and also R here. Now we go to the question 1, A1. Differentiate the rate of the pepsin enzyme reaction at temperature P and temperature Q. Okay, so you have to differentiate the rate of pepsin enzyme reaction at temperature P and temperature Q. So this is referred to the temperature P, this is referred to the temperature Q. Alright, so temperature P usually we start with the 0 degree temperature. 0 degree temperature. So you have to take a look on the rate of reaction. Rate of reaction on the point P here. Okay, when the temperature is 0 degree Celsius, and then this is referred to the temperature Q. So when in the temperature Q, what happened to the rate of the reaction? Rate of the reaction is at the highest. So you can predict. So this is referred to the optimum temperature. Optimum temperature for the pepsin. For the pepsin reaction. Alright, so temperature Q is referred to the optimum temperature. Optimum temperature for the pepsin. Pepsin reaction. Okay, so that is our early prediction about the temperature P and temperature Q. Okay, now let's take a look on the suggested answer here. So you have to draw the table with the 2 columns, temperature P, okay, temperature Q. Okay, so this is referred to the first mark, first point here. So first different, P, temperature P refer to the low temperature. Okay, while temperature Q refer to the optimal temperature. Okay, when both correct, you will get 1 mark. Alright, and then we go to the second point. The rate of reaction between pepsin and protein is low. Okay, the rate of reaction between pepsin and protein is low, or you just can write the rate of reaction is low. Yeah, the rate of reaction is low, right? Okay, low here. Yeah, we can see here the rate of reaction is almost no reaction or zero. Okay, and then Q, at temperature Q, the rate of reaction is rapid. Yeah, the rate of reaction between protein and also the pepsin is rapid. Yeah, very active. All right. Okay, next we go to the third difference. The kinetic energy of protein molecules and pepsin are low. The kinetic energy between the protein molecule and pepsin here are low. Okay, low because of the low temperature, whereby the kinetic energy at the of the protein molecules and pepsin at the point of temperature Q here are high. Yeah, the kinetic energy at temperature Q is high. Right. Okay, next we go to the next point or next mark. The frequency of effective collision between the protein molecules and the pepsin are low. Okay, the frequency of effective collision between the protein molecules and pepsin are high. Right? So the frequency of the collision between the protein and the pepsin are high. So it will cause most of the protein will bind, yeah, to the pepsin. Yeah? Okay, next. Only small amount of proteins is hydrolyzed by pepsin. Yeah, only a small amount of protein molecules is hydrolyzed by pepsin at temperature Q. Okay, while at temperature P just now, temperature P. While in temperature Q, high amount of the protein, is hydrolyzed into amino acid, by pepsin. All right. Okay. Next, last point is last formation of polypeptides. Yeah, last formation of polypeptides. Okay. Right. And then many formation of the polypeptides. Okay. All right. So you have to know that, okay, once the protein is hydrolyzed, it's not directly into the amino acid. So the proteins are hydrolyzed into the polypeptides, okay, by pepsin. And the polypeptides just now will be hydrolyzed into peptides. And the last one, the peptides will be hydrolyzed into the amino acid. Okay, so at temperature Q, less formation of polypeptide, whereby at temperature P, less formation of polypeptide. At temperature Q, many formation of the polypeptide. Okay, so polypeptide refer to the products. Yeah, refer to the products. Okay, so this is referred to the suggested answer for the question 1A1. Yeah, so all the full mark here is 5 marks. Okay, now let's go to the question 1A2. So explain the state of pepsin enzyme and the reaction of the enzyme after point R. Okay, explain the state. So underline state the pepsin enzyme. Okay, you have to explain about what? About the state of the pepsin enzyme. And also you have to explain the reaction of enzyme after point R. Okay, after point R, okay, so what happened? So you have to explain the state of the enzyme and also the reaction, of the enzyme. Alright, so suggested answer here, okay, point number 1, the temperature is rising. Okay, correct. So the temperature is rising. Increase. The temperature is increased. So the pepsin enzyme activity decrease rapidly. The reaction of the pepsin enzyme decrease rapidly. Decrease rapidly. So when the temperature is extreme, the pepsin enzyme is denatured. Okay, extreme more than mean that more than 60 degrees Celsius. When the temperature is extreme refer to the a temperature which is more than 60 degrees Celsius, so the pepsin enzyme is denatured. Okay, denature. Okay, what do you mean by denature? Means the chemical bond between the enzyme, between the enzyme molecule will break down and destroy. Alright, okay, then point number 4, the chemical bonds in the pepsin molecules are broken. Correct? Yes, okay, it will break down. Okay, then point number 5, the active site of enzyme changes. Alright, so why the active site changes? Because the chemical bond in the pepsin molecule are already broken down. Okay, then point number 6, the pepsin enzyme cannot bind to the protein and the protein hydrolysis is stopped. So the protein hydrolysis is stopped. So the rate of reaction will stop at extreme temperature, which is more than 60 degrees Celsius, because the enzymes are already denatured. Okay, so this is referred to the suggested answer for the question 1A2. Okay, we go to the question 1B. Okay, 1B. Diagram 1.2 shows a laundry detergent brand X. Okay, laundry detergent brand X. Alright, so this is referred to the washing powder. Okay, look at the description here. Contain protease and lipase, more effective than ordinary detergent. Avoid using boiling water. Okay, so this is the clue given to you. And clue given in the diagram. Okay, next we go to the question B1. Mrs. Salma choose brand X laundry soap instead of ordinary laundry soap, which does not contain enzymes. So justify the choice of Mrs. Salma. Okay, so you have to justify. So what is the choice of the Mrs. Salma? She choose a washing powder which is referred to the brand X. Brand laundry detergent brand X, which is biological detergent. Alright, so you have to give the points that support the the choice of the Mrs. Salma. Alright, so this is referred to the suggested answer for the question B1. Okay, first point, detergent contains biological enzymes. Okay, you have to state that this detergent contains biological enzymes, which are, okay, you can write the example of the enzyme which are referred to the protease and lipase. Okay, point number 2, protease. Okay, so what is the function of the protease? Okay, you have to state the function of the protease. Okay, protease enzyme can remove protein stain easily, or quickly. Whereby, okay, in the point 3, lipase. Okay, lipase enzyme can remove fat and oil stain easily or quickly. Okay, now point number 4, clothes are washed using low temperature or warm water. Clothes are washed using low temperature or warm water. Okay, why we have to wash in the warm water? Because warm water refer to the optimum temperature, that allow the enzymes act or function very efficient. Okay, then point number 5, more energy efficient or saver. More energy efficient or saver. Point number 6, the chemicals in the detergent powder are replaced with enzyme. So the chemicals in the detergent can be replaced by enzymes. So the enzymes in the detergent powder can be biodegradable. Can be biodegradable. Refer to the easy to degrade, easy to be break down. Alright, okay, then point number 8, does not cause water pollution. Right, so does not cause water pollution. That is referred to the point that you have to support yeah, support the choice of the Mrs. Salma. Okay, that is uh, the correct way how to answer the question that need you to justify. Yeah, so you when the question asking about you justify, so you have to give the point that support, yeah, that support the choice of the in the question. All right, okay, that is equal to the question 1b. Okay, next we go to the question b2. Mrs. Salma found that her silk clothes, yeah, was spoiled when washed with brand X detergent. Okay, explain why. All right, so silk clothes. All right, so you have to know that silk is made up of protein. Okay, protein. So when Pon Salma, yeah, wash her silk clothes with a brand X detergent. Okay, so in the brand X detergent there is a protease. Okay, there is a protease. So what happened when there is a protease? So protease will break down the protein. Yeah, will break down the protein. Okay, so this is referred to the suggested answer. Point number 1, silk is made up of proteins. Okay, point number 2, protease enzymes will hydrolyze silk protein. All right, okay, protease enzyme will hydrolyze silk. And point number 3, the protein structure will break. All right, so the protein structure will break. So that is referred to the suggested answer for the question B2. Okay, so that's all for the chapter 5, yeah, metabolism and enzymes. Okay, I hope you can study more about this topic, and please answer more question, yeah, more question to make sure that you really understand on this topic. All right, so see you again in the next video. So take care, everyone.